You found the skeleton. How would you calculate the dinosaurs? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hello, everyone who is here. It is I, Jackson. Welcome. Today we are talking with Bread of Life, Rebecca Davis, and we're also going to have Dapper Dinosaur on. So welcome. And Peter, we can show them. And thank you, Peter, as always, for producing this. Where would I be without you? I do not know. Hello.
Doing better?
Hi.
Hello.
Okay, so Rebecca. Are you fine with Rebecca, or would you prefer Bread of Life?
Oh yeah, of course. No, Rebecca.
Well, bread. All right. I'm also bread. I'm wheat. So
Wait, are you two
There's
secretly
connection
cousins?
there.
Are
we
you two
could be.
secretly cousins?
It could be. Could be.
Okay.
I've only met one other wheat individual who is not related to me. So entirely possible. Alrighty. So we are here to discuss evolution today, and specifically, at least to start, we are going to discuss a study, but I guess a study about Chlamydomonas evolving multicellularity. But I suppose before we jump into that, a little bit of background. I first heard about your channel a few weeks ago, Rebecca, and that's when I got in touch with you. That's when I sent you that first email. And so one thing I was interested in, so as not to strawman you or straw woman you, I guess, was to send you a list of questions, attempting to suss out where exactly we agree and disagree with regard to evolutionary theory. Straw person, yes, gender neutral. And if I may read those questions, because you did agree, you said yes to all of these questions, at least at the time of that I sent this email or that email. So if I may, I'm going to read the questions. So the questions that I sent were, do you believe that all organisms are born with inherent variations which occur through such processes, should have been processes, as mutation and recombination? Do you believe that the effects of those variations can be either harmful, neutral, or beneficial to their host? Do you believe that there exists a limited amount of resources in any given environment? Do you believe that organisms must compete with each other for resources? Do you believe that males and females can choose each other based on physical or behavioral characteristics? Do you believe that this process of competition for resources can lead to the fixation of particular variations within a population? And do you believe that the fixation and accumulation of variations in separate populations that share ancestry can lead to those populations becoming unable to interbreed with each other? And as of now, do you still agree with all of those points?
Yes.
Okay, so my question then sort of becomes, why don't you accept evolution as a whole? Because that's natural selection, mutations, natural selection, sexual selection, speciation. That's all of like the major processes. Obviously, it's not like endosymbiosis, genetic drift, and some of the other more recent and processes of evolution. But
Jackson, if I answer that, we're going to go back and forth, and we're not going to talk about the topic that we agreed to talk about tonight, which is this article that you sent me, De Novo Evolution of Macroscopic Multicellularity. So I just want to be clear, that is the article that I came on to talk about.
Yes.
And I'm going to be reluctant to enter into other types of conversations, and I told you that by email. And that's simply because you actually have so much more knowledge about this topic than I do. I am a lay person when it comes to this. So if we're talking about a topic, you're always going to have a natural, more information than I do.
I don't think
So
that's necessarily
I'm
the case.
reluctant to get into a conversation that doesn't include looking at a particular scientific paper or a particular topic that we've agreed to talk about.
Yeah, no, we are absolutely going to discuss this paper. I've prepared like a slide and went over the paper again and everything, so we can talk about this, and I can show the people who are watching like a little visual representation of this. So yes, I'm prepared to talk about it, but it is also germane to the process of evolution as a whole, because what you essentially asked for me was to show you something new in evolution occurring. Yes.
I asked you
correct?
to show me a change, proof that an organism can change to evolve a new system, a new organ, something like this.
Okay, and so that's precisely what happens in this paper. This paper occurs. Peter, if you can show the evolution process slide 24, please. So
could I ask a quick question? Because
sure.
I feel like we're in danger of having a little bit of talking past each other. When you say a new organ or a new system, Rebecca, what do you mean by those things? Because I've heard people claim that what I would consider to be a system isn't a system, or that what I would consider to be an organ isn't, and vice versa. So what do you mean by that?
Well, it doesn't really matter for this conversation. And like I said, I prefer to stick with talking about the article that we agreed to discuss, because your question could take us on a long rabbit trail and will probably involve me saying things that you're going to then push back against. And so I would prefer not to go there. Let's just, if you know, I have my Bread of Life channel is my primary channel, but I have another channel that is called Examining Evolution. And, you know, I'm inviting people to show me the evidence for evolution. Why should I believe this story that you guys believe? And so I've asked Jackson to present me with some very good evidence for evolution. He presented me with this article. So that's what we agreed to discuss tonight, is that we're going to look at this article and see what it shows us. What does this demonstrate about evolution?
Indeed. Yeah, and perhaps we can use that as a I don't know, because you want to just spend the whole conversation just talking about this one article, because there were other things you mentioned in the emails which I found very intriguing, which I would like to discuss additionally after this, of course.
Well, Jackson, like I'm slightly frustrated that we're having this conversation right now.
That's fine.
Because
that's fine.
we already talked. We had 25 messages back and forth before this conversation, and I specifically said I want to stick to the topic that we've agreed to talk about. This
also mention others, but that's fine. We can talk about this. All right, Peter, put up slide 24, please, from the evolution PowerPoint. And so if you put up slide 24, then that should show. Yes. Okay. Okay, so what we're talking about is the picture on the left here, the Heron et al. 2019 paper, which if you are a frequenter of my channel, you have seen me discuss this paper in the past. We discussed it in, what was it? I think it was Misunderstanding Multicellularity. I also made reference to it in Experimental Evolution Part 2. So you may be familiar with it if you watch my channel. So what in essence happened in this paper was the researchers, Heron et al., they subjected Chlamydomonas reinhardti, which is an ancestrally unicellular algae. You can find it in nature or order it from various companies, and it's unicellular. The only time at which there's more than one cell in a single body is when they're doing reproduction, and then they split and they go off their own way. So what the researchers did in this experiment was they took multiple lines of Chlamydomonas, and they subjected it to predation by Ochromonas valencia, which is a paramecium. And so, in essence, what this caused was there's a limited amount of space, and there's a predator, and so this caused adaptation in the Chlamydomonas, which was multicellularity. They developed extracellular matrices, and then they began sticking together. So they basically were undergoing clonal, or they were developing clonal cells, and multicellularity is all about clonal cell divisions. It's not about just sticking together of different genetically unrelated individuals. It is the sticking together of genetically identical or genetically very similar individuals. And so that's how you build up our cells. That's how you build up plants, fungi. Everything that does multicellularity is a clonal multicellular organism. So, okay. And so this took about 750 generations was how long it took to go from unicellular to ancestors to multicellular descendants. So with that in mind, so what were your thoughts on this experiment, Rebecca?
Well, actually, you're conflating two different articles, and the one that we're supposed to be talking about tonight is de novo evolution of macroscopic multicellularity, which has to do with
did
yeast.
I send you the? Oh, I'm so sorry. You're right. I sent you the yeast one. Oh my goodness.
Right. Now,
My
I
bad.
have read the one that you're talking
Sorry.
about, de novo origins of multicellularity in
I sent you
response
the
to
wrong one.
predation, about the
That was
algae.
my fault.
Now,
Yeah,
I
I
have
sent you the wrong one. It's okay.
Yeah. So I have read that one in the past, but it's been a little while. I actually made a video about that one, so you can watch my video if you want to see my response to the one about algae.
You can give it here
So
if you
that's
want.
on my Examining Evolution channel. So I actually read quite a bit of the article and discuss it.
Mhm.
But the one that I was prepared to discuss tonight,
I'm sorry.
is,
No, that was my fault. That was my bad. Yeah, Peter, go to slide 25. I'm sorry about that, Rebecca, and I'm sorry,
That's
audience.
okay.
I sent the wrong one, which means yes, Dapper, I did send you the wrong one too. So my apologies.
Jackson, when will the disappointment cease in our friendship?
Never.
Okay,
Dapper, I'm still
as long
here.
as we have it established.
Yes, this one by Bosdag et al. 2021. Yes, that's the one I sent you.
yes,
Right.
exactly.
Okay.
Yep.
Yes.
Now,
Okay,
I want to refresh myself. This is the one where there was gravity settling used as a selection mechanism for the
yes.
yeast, right? Okay.
Yeah.
Just making sure I know which paper we're talking about.
Yes. Yeah. I'm sorry. Yeah. Again, my apologies, people. So yes, in this experiment, there was selection by gravity. They basically clipped the ACE2 open reading frame, and this allowed multicellularity to develop. And then they just kept selecting it, and under anaerobic condition, and only under the anaerobic conditions, the anaerobic yeast developed not only multicellularity but macroscopic multicellularity. And then there's they did an analysis of which genes were mutated and how the cells grew and this and all that. So yeah, researchers grew 5 populations of ancestral unicellular, anaerobic, aerobic, and mixotrophic. That means they used both Saccharomyces cerevisiae. And after some 600 cycles of selection by gravity, about 3,000 generations, the anaerobic yeast became macroscopic, and they reached about 1 millimeter in diameter. Okay, so yeah, my bad. So with that in mind, with the correct one in mind this time, Rebecca, give me your thoughts, please.
Yes, well, let me tell you up front. I read this a couple of times today, and
Okay.
I'm not going to say I fully understand it. So I have questions to ask you about some of the things, like what does this mean? But I'll just start by asking you, just from something from the introduction stuck out to me, and I just want to see if you agree with this statement made in the introduction. It says, however, little is known about how nascent multicellular organisms consisting of small groups of undifferentiated cells evolved to form biomechanically tough macroscopic multicellular bodies, and whether selection for size itself can be a driver of multicellular innovation.
Yeah, I agree with that.
Okay. So then as we talk about this, right, we're doing it with the understanding that little is known about how this happened, if it did happen.
Not this one. They're not saying this experiment. They're talking about in general.
Yeah, of course.
The transition
Yeah.
from microscopic to.
What does this mean? In the absence of genetically regulated multicellular development, however, we do not know how or even whether nascent multicellular organisms can evolve the increased biophysical toughness required for evolution of the macroscopic size. I don't really understand that statement. What does it mean, genetically regulated multicellular development?
Well, and I am opening the article again. Are we opening the article? And this is in the
And that's
the intro or
line
the
49.
abstract?
Line 49.
Oh boy. No, not that one. You said this. Well, read it one more time for me, please.
Yeah, line 49. In the absence of genetically regulated multicellular development, however, we do not know how or even whether nascent multicellular organisms can evolve the increased biophysical toughness required for evolution of macroscopic size.
Okay.
So I believe
start.
I can help
Yeah, go
kind
ahead.
of
Okay.
translate this. So when we have genetically regulated multicellularity, you actually have a set of developmental genes that are telling the embryo essentially how it's going to develop. So at the most basic level, most multicellular organisms have at least one axis. They have, whether it's a top-bottom axis or a mouth to the opposite end of the body, and that's set up by these regulatory genes. And so without this, you can get microscopic multicellular organisms that have very basic divisions of cell types. Basically, it just comes down to like somatic versus reproductive cells. But without any more advanced regulatory developmental genes for, actual macroscopic development, it's hard to see how these microorganisms could then go on to develop into structures that are physically tough enough to be really big from the standpoint of microscopic versus macroscopic.
Yeah, if you the preceding sentences, you're right. Extant multicellular organisms have evolved a number of developmentally regulated mechanisms for increasing biophysical toughness or reducing the rate at which stress accumulates. Yeah, so basically what Dapper just said.
So we're trying to show if this toughness, if we can find a way to acquire this toughness.
Right, or how does it occur?
That's one of the...
Yes.
Yeah, if or, can it occur? How does it occur? Yes.
Which, while it might be a topic for another time, there actually have been further experiments past this with yeast that are showing ways in which multicellular yeast actually can increase in toughness without significant additions of genetically regulated multicellularity. So there actually has been further work since then, but since that's a different papers, we probably shouldn't go too far into it. But it is interesting that that has continued.
Okay.
Yes. Right.
Cool.
Sorry,
So
I had to
one of
find
the
a PDF of it because I didn't have it offhand.
And I'm just kind of going through in order in the paper at just some points that I thought were interesting.
Yeah.
And so one of them is that it is page 5, line 80. So we saw that, I mean, what they're describing in this is that with the yeast that had aerobic metabolisms, right? They did not get bigger in this experiment.
Or
and
the mixotrophic.
For people who are like, what is going on in this article? As far as I can tell, what happened in this article is that they played around, and you guys can clarify and change it up. Tell me, detail it or tell me where I'm understanding this wrong. But basically, they took some yeast and they played with it genetically. They took out, made it so that the gene responsible for aerobic respiration was no longer there. And they did some other tinkering as well, but that's toward the end of the article, so I won't mention that right now. But they tested basically 3 different populations, one with aerobic respiration, one with, I guess, what was the word used? Mesotrophic?
Mixotrophic, yeah. They did mixotrophic, anaerobic, and aerobic, yeah.
Yeah, and so the anaerobic, the ones that they tinkered with to take out their normal respiration, those like basically clumped together,
No.
got bigger and
One part.
tougher. Okay, explain where I'm
Okay.
getting that wrong.
So they didn't just clump together. These are clonal cells. So it is an individual growing, and they are
Yes.
continuing to grow, right? So
Yeah,
they're not
and-
just clumping, it's one growing.
Yes, and I
Which
should
then
have
becomes
been clear about
intertwined.
that. It's actually, they noticed that there was no aggregation, like it wasn't just
Yes.
cells that aggregated.
Yes.
It was cells that, you know, they did budding, and
Yes.
it was all from that same, like, okay, clonal.
Yeah.
So,
Mhm.
but they were able to like
They intertwined.
grow-
Yeah.
yeah, and so they basically formed a strong bond.
Yes.
So am I understanding correctly, like what happened? Is there anything you guys want to add to that?
So do you understand why they are doing it in these multiple different metabolic types?
Well, it seemed like they wanted to see what would if they could produce you know, organisms that would grow larger. They're trying to make it, they're trying to show how multicellular organisms could have become bigger, right? Because, you know, there's this issue with oxygen, and how do we get, you know, these, because of the dynamics with oxygen, how do we get actually organisms to get bigger ever? Right. And so they noticed the yeast. So you're agreeing with what I'm saying? Like, I'm doing all right with my explanation. So the yeast that the normal yeast that has aerobic respiration is not capable of getting that big, at least. And that's what the experiment showed. It didn't get any bigger.
Right.
But the ones where they took out the, you know, oxygen issue, right? They did grow. And so what I thought was interesting, and this goes right along with what I just wanted to read, on line 80, it says all 5 populations of anaerobic snowflake yeast evolved macroscopic size with individual clusters visible to the naked eye.
Right.
In contrast, snowflake yeast capable of metabolizing oxygen remain microscopic, a result consistent with recent work showing that competition for limiting oxygen imposes a powerful constraint on the evolution of macroscopic multicellular size.
Yes.
And so my question for you is, go ahead. Did you want to say something?
Oh, just saying that's a reference to another paper by Bosdag, I think, written the previous year, which is like the double-edged sword of macroscopic, or of oxygen on macroscopic evolution. Yeah, go ahead.
So my question is, isn't this the opposite of what we need to happen? I mean, I really don't know that much about biology, but don't most large organisms use aerobic respiration? So don't we like have a problem here?
No, because they're much larger than these guys. The organisms which are doing so, what they're talking about is the transition from microscopic size to macroscopic size, which is in this case on the order of 1 millimeter, required anaerobic conditions. But up from there, at least as seen from the fossil record and geochemical analyses, and again from other organisms which are way larger, you need oxygen. So yes, you did need anaerobic conditions to get over the hurdle into macroscopic size. And then from there, you have oxygen, which helps you get bigger. Because and you understand like why we need oxygen for, you know, like what we're doing with oxygen, right? We're doing cellular respiration so we can produce more ATP from more cells, right? Or for anybody in the audience who's not familiar. So yeah.
Right. So if we take these, let's say these conclusions are absolutely, you know, universalizable. So this is just how it seems life works in terms of getting to these larger size. What that would mean is that in terms of the history of life, the time where we start to actually get millimeter scale organisms is probably before the great oxidation event. And that's all that means, which is interesting, but it's it's not exactly like, shaking the foundations of modern biology or evolutionary history or anything. It's just
Yeah.
like, oh, okay, that's neat.
Okay. So then I guess I'm just kind of skipping quite a lot.
Okay.
But I wanted to talk about some of the methods that they use. Oh, where's my one page that I starred? I don't want to skip that. Hold on. Yeah, I wanted to see what you guys made of this. Okay. This is on page 15. It's line 278. It says,
Okay.
to our knowledge, this is the first long-term evolution experiment to directly show how simple multicellular organisms evolve increased complexity.
Okay.
Can you explain what that means?
So most of the other experiments that involve multicellularity, or if on sort of in this similar vein, are from unicellular organisms to multicellular organisms, like the one I mistakenly mentioned first. Or actually, so this work, one of the co-authors is William Ratcliffe. And so this work is in part built on a previous study he did back in 2012, which is how they figured out that it's the ACE2 open reading frame, which is involved in the switch from unicellular Saccharomyces to multicellular, to where they're sticking together and they're budding. They're doing the clonal budding. Right.
Now, I want to say something about that one since you're mentioning it. And that is, why did they pick algae to do that study? It's because algae is known to do that. So
well, those
nothing
are Saccharomyces is a fungus, not an algae.
Oh, okay. Well, then maybe we're not talking about the same one.
If you're talking about the heron et al., that was an algae. That was Chlamydomonas, but Chlamydomonas is also ancestrally unicellular. So no, it doesn't. It doesn't just make little clonal buds like that. That's not a normal phenotype that you observe in nature.
Okay, well, maybe we can look at that one another time.
Okay.
But I just wanted to say to the audience too, this paper is dated August 5th of 2021.
Okay.
So if this is a true statement, this is pretty startling that August 5th, 2021, this is the first long-term evolution experiment to directly show how simple multicellular organisms evolve increased complexity. Now, the reason I think this is important to say is because whenever I get in conversations with a bunch of atheists who believe in evolution, you're
making assumptions here.
Well, I have many Christian friends that believe in evolution. God bless them. I love them. But so, okay, we don't have to go there with the atheism. But
Thanks.
But they try to make it sound as if we just have it all figured out. So, that we have so much overwhelming observational experiments. We have so much empirical evidence for every step of the process. And that is the way that the picture is painted. So this is not my statement. This is coming from scientists who work in this field. And they are saying here that this is the first long-term evolution experiment to show how simple multicellular organisms evolve increased complexity. So at least on this topic, we don't have it all figured out. Is that right, guys?
No one would ever. I think it would be absolutely foolish to say we have everything figured out. We don't have everything figured out about natural selection. That was the first mechanism of evolution.
I think it might be handy to use a much less well-supported part of science that I still think people generally agree with, which is that we've, generally speaking, when you're talking to about science and stuff, we tend to say that we have a pretty good grasp on things like how gravity works. When in fact, there are circumstances, for instance, for very, very small things where we actually just don't have a clue. But that doesn't mean that we don't have overwhelming support for the parts of gravity that we, in fact, do have a lot of confidence in. So, for instance, we know exactly where Mercury is going to be in 3,000 years, 2 days, and 14 minutes, right? Like, it doesn't, no one's worried about this. On the other hand, yes, there are always areas, and even something as basic, we think, as gravity, where there's active research. We don't know what's going on in certain areas. Evolution is the same thing. We have overwhelming evidence for things like universal common ancestry, natural selection, sexual selection, genetic drift, recombination,
Speciation,
mutation,
all that.
speciation, all of this. But there are always going to be areas where we have things are under researched or they've been hard to research, and we're just now getting to those areas. So
epigenetics, for instance, it came out in like the 90s. You know, this is a brand new field within biology.
So the fact that until very recently we didn't have a lot of great research on this particular topic, that doesn't really do anything to counter the idea, say, that civets and dogs share a common ancestor, and they also share a common ancestor with horses and humans. Those are not really the same question. And while this is a really cool thing to talk about, even if we never figured this out, it doesn't get rid of the other things that we already have figured out about evolution, which is substantial.
Yeah, I have to agree with Dapper in that it's just saying this is the first that we've done of this. Okay, that is an observation, but it doesn't really do anything to the results of this study.
Okay, well, we can look at what the results of this study actually show, because I was, I'm surprised that you offered this up as, a strong example of, evolution that we've observed.
Yeah, because these guys sequenced the genomes of the progeny and figured out where the point mutations occurred that caused these changes in toughness and multicellularity, all that stuff. And actually, we can go back to the slide at some point if you want to look at those.
Sure.
But that's why I think that. That's why I personally think this is, even though I gave you the wrong one, so my fault. But yes, I do think this one is also a good study. Yes.
Well, I want to talk about then the results, and another startling statement they kind of make about this, and me knowing almost nothing about yeast, it surprised me to kind of see this statement. And this is on page 16, line 299. It says, who's crunching? Dapper Dinosaur, are you eating dinner? Are you eating chips?
No, I'm muted. I've been muted this whole
that
most
must
of this
be
time.
Peter eating chips back there.
Yep.
Yes.
Whoops.
Okay. So it says, our results depend on the fact that snowflake yeast grow as topologically structured groups with permanent cellular bonds. And we would not necessarily expect similar biophysical exaptation in organisms with alternative means of group formation.
Yeah, I agree with that.
What does that mean?
So
Jackson?
what that means is there are organisms who don't form these permanent cell bonds, and so they are unlikely to ever form multicellularity. That's why multicellularity has formed in a limited set of organisms. It's not in all eukaryotes, or well, it depends on how you define it for prokaryotes, but only some of them have developed this, but not others. Why? Right. Probably because of stuff like this.
Right. So they're depending on the fact that yeast already do this.
No, they don't do.
They depend on this
specifically. They have characters like permanent cell bonds, which can be accepted for this new function. It's not that they're not growing clonally, forming these snowflakes in the wild, at least from what anyone has seen. But they have characters which can be adapted for new functions. That's what acceptation means.
So what does this mean, though? Because I admit I don't fully understand this statement. Our results depend on the fact that
that's what I just said.
Yeast. No, I know, but you're not actually responding to like what this phrase is. That snowflake yeast grow as topologically structured groups with permanent cellular bonds. What does that mean?
That these organisms can develop these permanent bonds between their cells.
Okay.
They have the ability to do this. If you
Right.
didn't have
So
this
it
ability,
doesn't say
you could
as
a
a
priori-
result of us tinkering around with them. This is something that it seems to me they're saying
not
this is
necessarily.
this is what yeast do.
No, actually, they don't form these structures in the wild. We know because you can go look at them. The only way that yeast in the wild interact is they do something called flock type aggregation, which is where it's a whole bunch of genetically unrelated individuals. And they basically form a biofilm, but they don't form clonal yeasts. So they're saying the fact that they can form these permanent cellular bonds, which not all organisms can apparently, is part of the reason that they are able to do this, that they are able to form these snowflakes, because they can form permanent cellular bonds. Does that make sense?
Well, maybe we can read a little bit more of the context to try to
Go ahead.
see what's exactly being said here, because I'm hearing a little bit of difference between what you're saying and what seems to be expressed in the article. So going on, it says,
Okay, go ahead.
well, you're saying they don't normally do it, right? And I don't know.
Yeah, yeast I
have this and I have you. So I'm just trying to figure out.
Saccharomyces is an ancestrally unicellular yeast. They can make biofilms, but that is flock type aggregation. That is not clonal multicellularity. What they are saying in the paper is this yeast grow, with these permanent cellular bonds. Yeah, because they can grow with these permanent cellular bonds. We would not necessarily expect similar biophysical acceptation in organisms with alternative means of group formation. Yeah, if they had a different mechanism of group formation, then they wouldn't be able to form multicellular structures.
Okay, but you're saying this never happens like naturally, not in it. What is topologically structured groups?
So that's kind of using a bunch of math jargon, but the idea is basically that you have this emerging actual pattern to how the organism grows that isn't just a blob. That's why they're called snowflake yeast, right? Because they form these little like crystal-like patterns, and so that's topologically structured. And I will say I do think it's a needlessly obtuse way to say it, but there it is. Okay. Scientific papers are like that.
Well, I'm still hearing a difference between what you're saying, Jackson, and what the line seems to say. And maybe it's the paper being unclear, but it does say the results depend on the fact that snowflake yeast grow.
If you scroll to page, if you go to page 9, because I just happened to Google TOPOLOG, they said surprisingly individual macroscopic snowflake yeast were not composed of a single topologically connected component like their ancestors. They analyze the chain topology, but maybe they could have phrased that better, perhaps, or maybe that's just the way that they thought was best.
Okay.
Of the 5 lineages that independently evolved and this is right below what you cited. Of the 5 lineages that independently evolved complex multicellularity, all but animals possess permanent cell-cell bonds, and early multicellular lineages in each are thought to start out as simple, topologically structured networks. So they have this structure. They have this growth structure, which allows them to adhere in particular ways. Yeah, and they have cell-cell adhesion, which is permanent, and these are allowing them to be evolved for multicellularity.
Maybe I can add a few comments and maybe clarify what's kind of going on here.
Sure.
So there's a difference between colonial multicellular organisms. Colonial single-celled organisms and multicelled organisms. Now, for various reasons, a lot of single-celled organisms find it useful to basically link up with other members of the same species. Sometimes it's basically they have better chances of evading predation if they're in a group, or it might be that they have a better chance of getting food. So, for instance, if a bunch of not clones but related algae can clump together, they might have a better chance of staying, say, I don't know, near the top of the water column because of like there's updrafts in the area, whatever. Now, these groups already, because they have mechanisms for cell-to-cell bonding, already have mechanisms that if they become a clonal multicellular organism, is useful. Because while they weren't originally using this ability to connect cell to cell for multicellularity, now that multicellularity is being favored, where it's everybody's going to have essentially the same genome instead of just loosely related organisms, this comes in handy. It's sort of like how in the standard story of evolution, what usually has become wings in various organisms that have learned how to fly are things like legs, right? So bats started out in the standard thing for evolution. Bat wings started out as forelimbs. Same thing with bird wings, pterosaur wings. It's because these are already handy things. They already have muscles that go up and down. There's skin that you can stretch between fingers for additional lift. Similarly, with these single-celled organisms, this ability to form non-clonal colonies that are still single-celled, but they're groups, helps them all along the way to becoming truly multicellular, where you have clonal organisms, and then eventually you have things like cell types and macroscopic structures.
Okay, well, I don't want to get too hung up on this, but I will just go back to the abstract about, what is actually trying to be shown in this paper. And I don't think, what's being said here is that we are showing how these unicellular organisms became multicellular. That's not what's being shown.
That wasn't
here.
This study. No, that was a previous study that this is building off of.
Okay, but
that was the Ratcliffe et al. 2012 paper.
Well, so are you saying they took their multicellular evolved yeast and then did this experiment with that?
Yeah.
Because
so
what I'm seeing is that they just took normal yeast.
No, they did take. That's what the snowflake yeast came from originally. Was that was why they deleted the open reading frame, because when Ratcliffe et al. Did the previous experiment back in 2012, and they did the same thing, they did selection by gravity on these unicellular yeast. They developed the snowflake phenotype, which resulted from deletion of the ACE2 open reading frame. And so the researchers took that data and they said, okay, let's try this, but let's see if we can make them larger, basically. And so that's how this came out of. So that's why at the very start they talk about the deleting
how
of the
they tinkered
ACE2
with them to make them anaerobic.
Yeah, so the anaerobic and mixotrophic, that was separate, but yeah, they took what they knew to make the multicellular first, which they determined from the last experiment, and basically started with that, and then went from there. Yeah, and then put them in particular environments where they made them anaerobic or aerobic or mixotrophic. Yes.
But what I'm showing from here is that what they're actually trying to do is show the growth, right?
Mm-hmm.
That this is focused on the size of the yeast,
right? And the biophysical toughness of them. Yes.
Yeah.
Mm-hmm.
So, what was interesting to me is that, if you look in the materials and methods,
mm-hmm.
and on page 18, looking at, it's kind of a long paragraph to read. I can read it, but they did more tinkering than just with the anaerobic respiration. They also tinkered around with it to try to get, let me just read it.
Also, see it says here, we started with a unicellular diploid yeast strain. So they were not ancestrally multicellular. It's on 331.
Yes, but the question is, do they naturally join together in certain situations?
And that's what the flock type aggregation was versus the clonal aggregation or multicellularity that they are discussing in this experiment.
Right. So even in the wild, because yeast are wild organisms. They're all over the place. They're just in the air sometimes.
Mhm.
In certain circumstances, relatively unrelated yeast, so not direct descendant, ancestor type, or sisters, whatever.
Mhm.
We're talking like distant cousin type relationships, will link up, but they do not do things like form like a sort of like a labor sharing thing. Like they don't do like, okay, I'm going to be the structural cell, you can do the reproduction. They don't do any of that.
They're just
They're
linked
not
by
creating clones.
their surface proteins.
Right. So this is a thing that happens in nature. In fact, you can watch it happen. You can put some yeast in like various like water of various temperatures and whatnot and watch different things happen. One of the things that can happen under certain conditions is you can see yeast actually settling out of solution over time. And in macroscopic little dots. This is because relatively unrelated yeast are connecting up for whatever purpose yeast do that generally. But in this experiment, where the experimenters are inducing this to happen in such a way that they actually start to grow clonally. So you'll get a single yeast cell, which then will start to reproduce. And instead of scattering like normal, and then occasionally coming back together with relatively unrelated organisms, they don't scatter. They form a ball at first. And then in the case of these snowflake yeast, as they're very elongate cells, they start to form these little chains that come out. And then eventually, over the course of this, there's stuff with what kinds of respiration they're allowed to do because of the tinkering and whatnot. But either way, they're still going from a group of unicellular organisms that sometimes form colonies to a group of multicellular organisms that use the mechanisms that were used to make colonies to instead hold their own bodies together.
Right. And from what I'm understanding, the reason that they're able to do that is because of the anaerobic respiration, which would be too, like if they were, the normal aerobic respiration, they would not be able to stay together like that after budding and things like that. Is that right?
Yeah, hence why they stayed microscopic in the
but they did become multicellular in the aerobic lines.
Oh yes, they were multicellular in all of them. They started out with everyone being multicellular.
They just couldn't get as big using the aerobic respiration. And there are proposed mechanisms for this. One might be that, like, if you have to diffuse oxygen across the entire body of the organism, there might be a size limit imposed. If you're an obligate aerobe and you can't get oxygen to everyone in your body, every cell in your body, well, that's gonna be a problem. Like humans have that problem, right? That's what drowning or suffocating is, right? You can't get enough oxygen to all your cells, so they start to die. So in a case where you're just relying just on diffusion to get enough oxygen, there might be a limiting factor there. That's one possibility. There are other possibilities that are, talked about. And I mean, look, I'm not an expert on this paper. I don't have it memorized. But basically, while oxygen might impose this barrier, anaerobic respiration, which is slower and generally speaking less efficient, but it doesn't have that constraint that oxygen might have, where you need the oxygen to be available, and it might not always be available. And that allows these organisms to grow a bit bigger and cross the threshold from being like micrometer scale to being millimeter scale. Because remember, in this experiment, they got to be 1,000 times bigger than they started out with when they were still the microscopic snowflake yeast. That's a pretty significant thing.
Yeah, but now let's read how they got them to do that, because it also did not happen naturally. Even after they tinkered with the genetics and made them not able to respirate, now they
so they selected or they took out the for the initial clone of the ACE2 snowflake yeast, we selected a due to a large deletion in its mitochondrial DNA, the snowflake yeast is unable to respire. It is metabolically anaerobic and was cultured in this media. And so then they selected them to selection by gravity, so that we evolved 5 replicate populations of mixotrophic snowflake yeast, obligately aerobic and anaerobic yeast in this broth, growing them in blah 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 culture tubes. We use settling selection to select for larger size clusters once per day, blah, 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 blah.
Okay, so is that it happening naturally, or is that more human intervention to like unnaturally cause these things to cluster together more.
So they are just selecting them by gravity, and on their own, they are mutating their genes in such a way that they are getting larger. So that is totally natural.
This is gravitational selection, not natural selection.
This is selection. Yes, this is artificial selection, but we are still selecting mutations, which we can't control which mutations they're having, but they are evolving novelties while under the duress of this selection process.
Well, let's really look at that. I would like to see.
Yeah, absolutely.
And let's look at the results. Where are these novel things? Because I looked at the same things that you put up.
So,
and I did see point mutations, but I did not see.
That's where it's happening. They even specify which genes the point mutations happen. Okay, it's if you look on page 14, they have a chart with the genes evolved in cell cycle process, filamentous growth, and cellular budding. Yeah, whole genome sequencing reveals the dynamics of molecular evolution and the genetic basis of cell level and cluster level changes. And we go to E. Macroscopic snowflake yeast were significantly enriched in mutations affecting cell cycle progression and filamentous growth. In addition, we saw mutations affecting budding, i.e. the location of buds on the cell surface, and bud neck size. While not a formal geocategory, these constituted 12 out of the 99 total genes containing non-synonymous mutations in genotypes isolated from PA1 to PA5 after 600 days of evolution. So yes, that is mutations in the population, not human-induced, which are being selected.
Well, now you're saying not human-induced, but if you look at page 23, okay, we have on line 45, look what it says. To genetically engineer a snowflake yeast with cell lengthening and bud scar strengthening mutations, we first deleted each gene separately in the ancestral background.
Mm-hmm.
Okay, so it sounds like they're tinkering with more than
ACE2,
just a respiration.
That was this is the ACE2 that was referred to earlier. That was the thing they deleted first. They deleted the ACE2 first, which is what induces multicellularity, which they figured out in the previous study. And so to start, they had their unicellular yeast, deleted ACE2, and then for the anaerobic strain, they also deleted the mitochondrial genes, which is what made them anaerobic. So yes, they made them multicellular as a result of the last experiment, and made them anaerobic, and then selected them, put them under selective pressure, and whatever mutations they developed were on their own.
Well, I mean, it's a little strange because it says we brought all three mutations under the ACE2 background, generating a quadruple mutant. Finally, we quantified the cellular aspect ratio and cluster size of this mutant by imaging uncompressed clusters under Nikon. Okay, so it sounds like they're engineering all these changes. So
again, these are changes that these changes were the ones that were witnessed in the previous experiment. Because so what happened with Ratcliffe et al. Was they just selected unicellular yeast. I don't remember. As far as I'm aware, I don't think that they deleted anything. They just had these yeast and selected them by gravity. So which ones settle out the fastest. And what they found when they analyzed their strains was that ACE2 was deleted from the ones who attained the snowflake yeast multicellularity. And so taking those results, they said, okay, we'll just delete these at the start to induce multicellularity because we already know that's how that works. And then they did made them anaerobic or aerobic or mixotrophic. So that's what they're talking about there. That was the reference to the previous study.
Okay. Well, I mean, like we're referencing the previous study quite a lot, but if we just talk about what are the results of this study, I mean, if you want, like we can read the other article and do this again, because what I'm reluctant to do is to like instantly accept all the results of the previous study and all your conclusions about it when I
Well,
haven't read that article.
The previous study was that
Mhm.
snowflake yeast or the snowflake phenotype is induced when you select them to settling out, which does it fastest. And then they found that ACE2 or the deletion of ACE2 or inactivation, whatever you want to call it, was what resulted in that phenotype. That was
Okay.
the previous study. That's all. That's from what I remember. That's all they found. I mean, there may have been some other stuff that they found too, but that was the major conclusion. And so they're taking those conclusions because Ratcliffe is one of the guys on this study. He's in this. He's one of the co-authors.
Okay.
And so they're just taking the results and going further with the results that he had, basically, or that they had.
What is it about this article that you think presents me with evidence that evolution happened?
So evolution did happen because for one thing, we had the other article, but like I said, another conversation. So just taking
if I accept that article too.
I said just taking this article, just ignoring the other article, just accepting this one, what we saw was there were mutations which occurred in these organisms affecting cell cycle process, filamentous growth, and cellular budding. And all of these resulted in macroscopic size. And these changes went to fixation in these populations due to a selective pressure. The alleles changed in frequency as a result of this mechanism, of this process.
Okay, so were yeast incapable of cellular budding before this experiment?
Yes.
You mean?
Well, they were incapable of budding as multicellular organisms.
Yes.
Yeast already reproduced in a single-celled budding, but that's a very different kind of thing. That's where you actually start growing one extra cell, and then when it gets big enough, you finally kind of pinch off the cell membrane, and then it leaves to go live its own life. Whereas this kind of budding is an entirely different thing, where certain spots on this multicellular organism start to grow into a little ball with a connecting chain of cells, right? And then eventually that chain of cells is going to die, and that bud is now going to go off and become an entirely new multicellular organism. So while we call both of these processes budding, they're actually extremely different. So it can be a little confusing there. So wild-type yeast do not do multicellular budding. Instead, they do a single-cell budding that uses entirely different mechanisms in terms of things like what's actually happening on like a genetic and biochemical level. This is a very different sort of reproduction.
Yes.
Okay, so what about this article would supply me with a reason to believe the story that you guys are telling about human origins and origins of life?
Okay, so again,
development of life.
So for one thing, this is evolution in action. Again, to repeat myself, because there are mutations which occurred, which affect all these processes, which were not tinkered with. They occurred in the organisms, and then the researchers sequenced their genome, and these mutations went to fixation, or these alleles, went to fixation in a population as a result of a particular selective pressure. That's definitionally evolution.
How are you justifying the statement they weren't tinkered with?
The mutations that they acquired in for instance, the anaerobic strain. So they're anaerobic and they're microscopic snowflake yeast, and then they were just selected by gravity. So the mutations that they acquired during that time that they were being selected, what was it? 600 generations, I think is what it said.
I believe it said 600 days. I'm not sure what the generation time for yeast is, but it's probably less than a day.
Yeah, but whatever, however many. Actually, was it 60 days or 600 rounds, whatever. Anyway, after this time period that this occurred, or during this time this occurred, they were developing mutations, not being tinkered with, okay, according to the paper anyways. They were developing these mutations which conferred biophysical toughness and larger sizes, which was not present in the original population. So this is a novel phenotype built on another novel phenotype under a certain selection pressure.
Can you show me where in the paper it says that it was those mutations
Yeah, I literally
that
just read that.
caused no. Where in the paper does it say that the mutations that we observed were the cause of the toughness? Because I don't know that much about yeast, but I do use it for baking. And I know one of the reasons that we use yeast for baking is because it actually does adhere to the gluten and it makes a tough bond. And that's why you need for so long, it actually produces this entanglement with the gluten, which is why it's one of the reasons that it's very good for bread. So, and the more you knead it, the more it becomes, those bonds become secure. So what I'm saying is, I would like to know where in this paper you're getting the idea that those mutations that occurred were the cause of the toughness and the entanglement.
So, first, page 13, line 238, so very top. We tested the effects of cell lengthening mutations, AKR1 and ARP5, and a putative cell-cell connection strengthening mutation, GIN4, in the ACE2 ancestor. Together, these mutations led to a 7.7-fold increase in average radius, confirming that mutations affecting cell length and cell connection strength can underlie the emergence of drastically increased multicellular size in snowflake yeast.
All right, good job, guys. I give you that one. But I want to reread this just to make sure I'm correctly understanding it.
And right above it, it also talks about the other mutations which occurred. Oh, sorry. Oh, okay. So 3,000 generations. So whatever it was, 600 days, 3,000 generations. Gene ontology terms known to affect cell length were significantly enriched, namely genes of cell cycle and filamentous growth. In addition, we found 12 non-synonymous mutations in genes with known roles in cellular budding, which includes 8 genes which have previously been shown to increase the size of bud scars. So it talks about all those.
Where are you reading?
Bottom of page 12, line 226. So basically 226 is the paragraph right above, and it's just talking about the other mutations that occurred also, so additionally. But yeah, so the point with this, mutations arose in 2 of these different populations, indicating parallel evolution. All else equal, larger bud scars should increase the amount of cell wall shared between the mother and daughter cells, potentially increasing the strength of cell-cell connections. Okay.
Now, why does it say potentially?
Because that's how science works. You always use guarded language so that you do not make a claim that you can't support. Basically, you're never going to find a scientific paper that says, we proved that this mutation did this thing, and that's why they grew big. Because the thing is, if you are then shown to be wrong, there's always a chance, even with very, very high confidence results, there's always a chance that you might be shown to be wrong. If you made claims that you couldn't support, that's bad for your career. There's a very good reason that science always uses things like, this indicates, probably, most likely, the data seem to show. That's just how you write in science. What you really want to look for, if you really want to dig into doing a lot of scientific papers, is looking into things like how confident they are, what are the p-values that they're giving you for these things, where there are squared values for their statistical analyses. But the problem there is that those are the parts of science that humans are really bad at stats just naturally, so it's hard to understand those parts.
And they did give a couple p-values here, and they're pretty small. 4.68 times 10 to the negative 10th. It's a pretty small p-value.
Okay, what does that mean? When you say that's a pretty small p-value, what does that mean, Jackson?
Roughly speaking, and it's a little bit more complicated, the p-value is the chance that you got your results simply because of a fluke of statistics, and that it's not actually any kind of causal link between what you're experimenting on and the results. So, for instance, if I have a p-value, and this is an extremely high p-value, but if I had one that was 0.1, that basically means that there's a 90% chance that the results of my experiment are actually because of my hypothesis. And there's a 10% chance they're not, which would be very bad in science. You would not publish with a p-value of 0.1. Now, this is just a very rough definition. There's actually a much more rigorous mathematical definition that I can't recite off the top of my head. But
you also typically want p-values which are like less than, 0.05, isn't it?
Yeah, and this one is
Yeah.
1 times 10 to the negative what?
-10th. So
Yeah, so this is 0.00000000001.
which I think is
That's
less
the
than
chance. Yeah, that's
0.05.
right. That's the chance that this is just, and again, this is roughly speaking, the chance that this result is just a result of statistical noise. Just randomness happened, and now that we got this result. So then what you want to do is you want to repeat experiments like this. And if you can keep getting statistical results with such low p-values, essentially what you can do is you can multiply them together. And as I'm sure we all know, when you multiply an extremely small fraction by an extremely small fraction, you get an even smaller fraction, a much, much smaller fraction. And so as that time goes, your confidence increases. But because this is science, and science isn't in the business of proof like math, where we can become 100% sure, we're always maintaining and writing about what level of confidence. And we have various statistical measures, and p-value is one of the most common statistical measures of confidence in your statistics. And that's, again, comes back to the reason why in science writing, you don't use strong affirmative language. Like, we showed that for sure these genes caused this thing, which resulted in this phenotype. You say, these probably resulted in this, and here's our data, and here's our statistics. And this is our level of confidence, and that's why we conclude this is most likely the case. Because even if there's 1 in an 18 trillion chance that you're wrong, that's not being positive. So in science, you indicate that in part by your writing saying, probably, most likely, it seems.
Okay. Well, you guys have been using much stronger language than is in this. So why are you guys using stronger language than the paper presents?
That
'Cause I'm
doesn't
not
really
writing a
seem to be
paper.
an argument. But
Yeah, I'm not writing a paper. It's basically that it's a relatively casual conversation that is not likely to be assessed with a fine-tooth mathematical comb.
I don't think that the people who are going to like review my thesis are going to come back and be like, you said in this conversation. So
yeah, if I were preparing this talk for a peer-reviewed paper, like if I were going to somehow try to write up like a conversation with a person on the internet, which I don't know what I would publish that in, but if I were to try to, if that were my goal, I would be much more circumspect. And I think Jackson would too. But like, it's sort of like, let's say you're 80% sure that like you turned off the oven when you left the house today. And so when someone asks you, hey, did you turn off the oven? You just say yes. Are you actually confident enough that it would stand up to scientific scrutiny? No. But that's not the conversation you're having, is a scientific paper about whether or not you left the oven on. And yes, I'm using absurd examples. Just, kind of roll with it, please. Because, I don't know, I'm a goofy kind of guy sometimes.
Yep.
Okay.
Goofy guy.
Yeah, so for me, in my everyday speech, where I'm not trying to be, rigorous to the level of a peer-reviewed paper, if some data indicates that there's like a 1 in 100,000 chance that whatever I'm talking about is wrong, I'm just going to say that it's right. And then if I turn out to be wrong, I'll make a correction. Look, I've issued a lot of corrections on my channel. There's plenty of videos where you can see on my channel where I say, hey, I got this wrong. Here's the correct information. Sorry about that.
Yeah,
so I'm not afraid of making a correction.
And I do it in the comment section of whatever video I screw it up on. So,
Yep.
Yeah.
Okay. So is there anything else you guys want to say about this?
Well, I think
go
I
ahead,
have a
Pepper.
question.
Sure.
What about this doesn't appear to be evolution as scientists would generally define the term in terms of biological evolution? Because to me, it seems extremely obvious that this is evolution, but it seems not to be that way to you.
Well, I mean, this is yeast doing yeast stuff with a bunch of human tinkering that, is not giving it like a new feature, a new system. I mean, it's using a capability that it already had and doing something slightly different because of the way that humans tinkered with it. So how is that a natural process?
In nature, that's like exactly what happens all the time, minus the human tinkering. But yeah, like all those papers I sent you, which I kind of hand waved, they're all something tinkering But I don't think in most of those other examples they were just like natural, like the Ragavalia example, where a gene duplication happened. Or I think I sent a couple of gene duplications, like one, a couple of gene duplications. But the point is, these are happening like in the wild. Like people weren't running around like.
Oh,
picking
Jackson,
up,
you're breaking up a little bit.
Oh, sorry. Can you hear me now?
I can.
Okay, people weren't running around like duplicating the genomes of crawfish. This is just something that happened in the wild. Or the garter snake example was another one where a point mutation happened, which slightly changed the way this protein worked, and now it's not susceptible to a toxin that's in it's one of these
Right.
animals in its environment. And so that's
Right.
But those are all examples of something minor, something small, just getting slightly changed for some new purpose.
At the basic level, that's what evolution is it's relatively minor changes each individually, but over time many minor changes can add up to what seems to be very major changes. And really the difference in this experiment versus nature is just that this is a controlled experiment, and of necessity controlled experiments are the kinds of things that humans have to be involved with, otherwise it's not a controlled experiment. And so one of the things that concerns me is a lot of times creationists will demand that certain things be shown in a lab, but then when they are, they'll then just complain, well, that just means that it took intelligence to make it happen. And so it quickly becomes a damned if you do, damned if you don't when it comes to experiments in evolutionary biology and creationism, where literally nothing that could be shown will be accepted because there's always the post hoc excuse of either, well, you didn't observe it in the lab, or if you did, well, it took human tinkering. Well, we don't labs without human tinkering, unless we have aliens available or very, very advanced robots, is not likely. But even then, you'll just say, well, the robots did it. So I don't see this human tinkering as in itself a good reason to reject this, because everything else that happened in this experiment is just a tightly observed version of what we see happening out in nature, where organisms can acquire new abilities like resistance to certain toxins, or the ability to exist in an environment with a new average temperature, or becoming a little bit faster, or becoming a little bit stronger, or bigger or smaller. And so why isn't that? Is it just the human aspect that makes it not evolution?
Well, actually, the reason that it's not convincing for me is that all of the examples I see are breaking the house. Okay, we're introducing mutations to things that are present in the DNA, right? But it's like this yeast lost a capability, right?
And then it gained new ones.
Yeah.
Well,
it lost the ability to pair off the daughter cell from the parent cell, which is multicellularity. I mean, we could flip this. We could invert it. And actually, Dapper and I have joked about doing this before. Like, you could explain everything in evolution in terms of loss.
Right.
Like, fish evolved onto land. They just lost the ability to breathe underwater. They just lost their fins. Well, yeah, you could explain it that way, but that kind of misses the point, doesn't it?
Yes, there's literally nothing in evolution can't be explained in terms of what was lost in getting you there. But the thing is, overall, evolution is a trading game, right? You never get to be Superman. You're always saying, okay, I'm going to become worse at X because Y is more important in my current environment, and I'm going to become better at Y. And that's a favorable trade for this species. And then you go from there. And so in all of the history of life, going from the simplest single-celled organisms at the beginning of life, however that's going to get started, which is a different conversation, all the way to humans or big scary dinosaurs or shrews or beetles or whatever, there was always trade-offs. Every time something acquired new traits, it became worse at other things in the past. So for instance, in humans, right? As humans become smarter and smarter, they have to do things like care for their young for longer and longer. So they lose the ability to have gigantic numbers of offspring, right? Humans generally can't have, like 45, 50 children per human. Like, you just physically can't take care of that many kids for as long as it would take to raise 50 people that you gave birth to. Like, it's not going to happen. But on the other hand, they gained abilities to become better able to adapt their environment to their own needs, which was fantastically successful, as we can see with the current very, very high population of humans, which I think it's coming up on 8 billion, isn't it?
Mm-hmm.
So, but there are trade-offs. Humans also ended up becoming less strong, but it didn't matter because in gaining smarts, they were able to do things like fashion tools. Similarly, with the snowflake yeast, sure, you're losing the ability to quickly reproduce into single-celled organisms that can quickly spread through the environment, taking advantage of all the available food resources. That's a downside to being multicellular. But on the other hand, if doing that comes at the cost of the vast majority of your offspring simply dying, well, that's not worth it. It's better to be less efficient at getting resources in the environment if you can survive. And so that's what you do. And so that's all of evolution. It's always a trading game.
Well, I was referring to the loss of aerobic respiration. So, I mean, they took something away. And I mean, you guys can say that, oh yeah, it's all loss, it's all trading. But I mean, you're wanting me
It's not
to
all loss.
believe, you're wanting me to believe that your eyes, your heart, your brain all happened from this trade-off. Like from little,
Correct.
mutations. And that is a story I just don't buy.
Cool.
And so you've got to, I mean, you think about how many systems are in your body, your skin. You're in a compact space. I mean, you're jam packed with systems that are all working together in harmony, down to the cellular level. Your cells are working together in harmony.
I don't know if I'd say they're all
as a
working in
dinosaur,
harmony.
I'm 40% empty space. Lots of air sacs in dinosaurs.
Okay, but also
sorry, that was just a joke.
Yeah, but also, yeah, I mean, yes, but also no. I mean, lots of your cells don't work perfectly well. Lots of your organs or organ systems don't work perfectly well in tandem with each other. I'm not really sure what your point is. You're just kind of saying it's cool that this is all happening. I agree, it's really neat. But
yes.
And
are you joking? I mean, you're taking in air. Your lungs are getting the air into your heart and your heart's getting it into your blood. Your blood is delivering it all through your body to all the cells to bring nutrients to all of your body. They're not working together well.
You're literally just saying,
I mean, guys, come on.
I said not all of your cells. A lot of them do work. Otherwise, you would die. But cancer is a thing that happens also.
Yeah, and you guys want me to believe that we were produced by things like cancer.
No, actually.
Well, mutations.
Okay, hey, let's back up.
They're more commonly a very bad thing.
They're not actually.
They're almost entirely neutral.
Yeah, you have not clearly read any studies like by Chimera.
Well, guys, I mean, you've got to make them more than neutral.
That's true. We do.
That's not what you said, though.
How in the world do neutral mutations add up to make all the systems in your body?
I think you're purposefully mishearing what we're saying, because the first thing you said was that they're overwhelmingly deleterious, and our response was simply, no, that's not true. They're overwhelmingly neutral, to which your response was, then where is all the beneficial coming from? That's a separate issue.
Okay.
And we can address that, but that's fine. But
I also point out
issue.
I'll take your point about the neutral. That's fine. I'm fine with that. But
also, initially neutral mutations can in fact then lead to beneficial mutations. So, for instance, we've talked previously about things like gene duplication.
Yes.
Gene duplication is generally neutral because now you just have 2 copies of a gene that you already had. That's great. But what it does is it means one version of that gene. So you have 2 copies now. One of them can then go on to mutate into a new protein that may in fact be beneficial, while you get the old copy to still make the old protein. So one relatively famous example of this happened with some ice fish, which originally weren't ice fish. They were regular, nice warm water, well, cool, but not icy water fish. And one of their, Jackson, remind me, which gene was it that duplicated? Because
trypsinogen.
Okay, so their trypsinogen gene duplicates. Now they got 2 copies. One copy happens to have one of the rare but observed beneficial mutations, which actually turns it into sort of an antifreeze. It helps prevent water molecules from forming the strong hydrogen bonds that turn them into ice crystals, which means in this case, it can actually have super cool water in its body, but it still gets to make the old trypsinogen. So what started out as a neutral mutation was subsequently acted on by a mutation that ended up being beneficial. So you can, in fact, have buildups of neutral mutations that eventually become beneficial. Another example is the mammal placenta. A lot of the genes regulating the mammal placenta actually come from a virus that infected the ancestor of mammals before they were mammals. And one of the things that viruses like to do is suppress your immune system, because otherwise it's hard for them to get going. But also, pregnancy suppresses the immune system, because otherwise your body would attack the fetus, which would result in very low birth rates, like near zero. And so one of the things that this viral insertion, which was originally slightly deleterious, but then became neutral as whatever organism it was infected with managed to neutralize the virus. So now it's neutral. And then eventually it becomes partially reactivated to help suppress the immune system locally in the uterus to then help pregnancy occur, so that now mammals that are placental mammals, which is, like most of the mammals that aren't marsupial. So most of the mammals that you think of, dogs, cats, humans, bears, wolves.
I realized you were talking, or while you were talking, Dapper, that I did actually have a slide I could have shown for the trypsinogen thing. I'm sorry, I didn't even. I just saw Peter's message.
So, but this is an example of what's originally a negative mutation, because viruses are generally bad. It becomes neutral when it's inactivated. So now you just have this extra viral genome hanging out, not doing anything. And then eventually it actually then becomes beneficial. So we do actually see beneficial traits building up from what were previously neutral, or even in some cases slightly harmful mutations. Because remember, even a viral insertion, ultimately it's a mutation because it changes the genome. Any change to the genome that's going to be heritable is long-term going to get counted as mutation. Doesn't matter where it comes from. And so, yeah, we do see neutral mutations building up into eventually becoming beneficial. In fact, we also see that in the Lenski experiment, where we had the
yes, Peter, you can pull up slide 11. Keep going, Dapper.
Okay, so in the long-term Lenski experiment, we have these E. coli bacteria that are being put under
the evolution process, slide 11.
So they're being put under very heavy selection pressure, where they have only a little bit of food relative to how many bacteria are potentially in here. And they're in a solution where there's food and also citrate. And normally, E. coli can't metabolize citrate. But one of the lines that was being grown in this experiment eventually became able to metabolize citrate in an aerobic environment, which they couldn't do originally. But it required several primer mutations that themselves were neutral. And then once several primer mutations built up, just randomly, that's why it didn't happen in every strain, then eventually a beneficial mutation was able to act on all of those. And suddenly this strain of bacteria that originally couldn't metabolize the substrate suddenly could. And it was a very big change in phenotype that happened essentially literally overnight, because that's how the experiment works, right? They let the bacteria grow for a day or so, and then they strain off some of them to use in the next batch. Well, one day when someone came in to do this, they noticed that rather than being relatively clear, the liquid medium that they were growing the bacteria in was still cloudy, meaning the bacteria were still actively reproducing far along after the normal food source was gone. But again, a bunch of neutral mutations had to precede this. And you can actually see in the table here up at the top left, there's a whole big difference. Every single difference that you see, so you get the initial split between what's labeled RNK on the left, and now there's all this extra stuff in between that and what's labeled MA on the far left of the right-hand section. Every single little insertion there is its own mutation. And when all of them are in place, suddenly we have a beneficial mutation. The same thing, a similar thing, not the same thing, happened in SIV, simian immunovirus, to create,
I don't have a slide for that.
Well, to create
Peter, you can take the slide down.
HIV group 2. And group 1 essentially doesn't exist. I mean, there's like 12 known cases, and they're all from like Central Africa or something. The group 2 HIV virus is the one that's actually spreading, worldwide. And one of the immune proteins that is common to all primates is tetherin, but humans have a weird tetherin. It's tiny. And so SIV, the virus that circulates among things like chimpanzees and gorillas, has trouble getting rid of human tetherin. And so the human tetherin just kind of pulls the virus off SIV if you happen to be exposed to it, no big deal. But in HIV group 2, there had to be 4 separate mutations, which once they were all in place, allowed collections of a particular protein in HIV group 2 to then successfully antagonize tetherin, even though that was never the job of that protein in the first place. And so again, we have multiple point mutations that are required. And once we get the final one, suddenly we have an observed beneficial mutation. So it is true that neutral mutations can eventually build up until you get the right one. And we've observed it happening.
And the same thing happens with antibiotic resistance. Also, you have various polymorphisms, which are not in any way useful under normal circumstances because they're not coming into contact with penicillin or its derivatives or cephalosporin or carbapenem. But suddenly these point mutations become useful when they're introduced to an environment that has penicillin or one of the others. So yeah, just to add on to your point, Dapper.
So I feel like we've now just snowballed a whole bunch of examples, but I feel like we've given examples of what you were looking for, which was how we can use neutral or, in some cases, even harmful mutations to eventually arrive at a beneficial trait.
I know. I was familiar with most of those examples, and I didn't say that there's never a beneficial mutation.
Okay.
What I'm looking for is because I know that you guys believe this story and you believe it wholeheartedly, but for me, it's an absurd story that all of these organisms, the great variety of life that I see on Earth, all came from these processes that you're talking about.
Why?
So
What about because you agreed to all of the questions that I gave, which were the outlining steps of how evolution works, or at least the large part of it. And you didn't disagree with any of those.
Right. Well, because I all of those involved small changes. But what
Yes.
you want me to do is accept that small changes can add up to these gigantic changes.
Yeah. And we
And-
also have mechanisms for how that happens, like heterochrony, homeotic mutations. These are things that, again, are pretty well tested. And when you run phylogenies on different groups of organisms, you see the major differences are like heterochronic mutations or homeotic mutations. So yes, large
you know
changes
what?
can also happen.
I mean, Jackson, you're welcome to bring your evidence. I mean, you've sent me some other articles, and some of them were the referring to the ones that Dapper Dino was just discussing. So,
Peter, can you put up slide 19? Yes. Well, all the examples I gave were all, again, like relatively small because most creationists say things like beneficial mutations don't exist. So
Jackson,
that's why I sent you those.
This is your most adorable slide.
So these are referring to Hox genes, which are similar yet expressed differently between 3 different phyla, and those phyla are Arthropoda, Tardigrada, and Onychophora, and they are expressing many of the same Hox genes just in different ways. So the differences between them are really how you express particular genes. Okay, this paper is from this chart is from the paper on tardigrade homeobox genes, where they look at how it's constructed.
This is less for Rebecca and even less for the audience, just for me. Are we looking at ecdysozoans or spiralians, Jackson?
Shut up, Dapper. Shut up. Stop talking. But these are all ecdysozoans.
Oh, okay, good. I just wanted to make sure.
So funny.
Sorry.
But as you can see on the chart, chart A and B, both chart A shows where the Hox genes are expressed, and B does it also. But yeah, so it's the same Hox genes, just expressed in different ways, and some of which are neo-functionalized in these different phyla. But that's the major morphological differences between them. Do you think that that's impossible, Rebecca? Do you think that's impossible for mutations to change the timing of development of where these genes occur?
Guys, you're asking me something that's way beyond my expertise. I don't know.
Well, you said that you don't believe large changes can occur. So clearly you're making a statement on this. And I'm asking you, here's an example in which the large changes are caused by differential expression of these shared genes. So do you think that didn't happen? That's not the way that happened, or what?
Well, I don't think that I have enough information on this to say how it happened. So I mean, like I said, if you want to give me a paper or something, because
Sure.
you could introduce things like this all night, but
Sure.
I'm
I can send you.
uncomfortable with that format because you have more information than me, but I would like to see the the actual data and the research to understand whether I should agree with your conclusion about it, whatever your conclusion is.
Well, my conclusion is just what the paper's conclusion is. But yeah, I can send you the tardigrade.
Jackson, you're breaking up a little bit again.
Oh, I'm sorry. Yeah, I have terrible internet. I'm also opening other.
Well, I think one thing that is interesting to me is that one of the things that that slide shows is that nowadays we tend to think of the difference between phyla as being these gigantic gulfs of uncrossable differences, right? Like, my goodness, a different phylum, it's basically just completely different. But actually, when you dig down into the actual like developmental genes that produce these adult forms of organisms from what just starts out as a sphere of cells, right? What you actually find is that it's relatively
Excuse me.
bless you.
Sorry.
No, hey, look, bodily functions occur. We're all biological organisms.
I sent you the paper.
And it's all because of those mutations. I mean, I'm sneezing right now because of mutations.
Probably not. You're probably
well,
sneezing because of like pollen or.
Right, but the sneeze reflex is genetically coded.
Sure.
So, but anyway, so what you actually find that when you dig down is that even the difference between phyla comes down to relatively small amounts of differences in which regulatory genes are expressed when.
Yeah.
And this even goes not just with those organisms, which what Jackson was using, these were all panarthropods, which is a very big group, right? It includes lots of things. But even if you go between, say, arthropods and like chordates, some of the same genes code for some of the same things. So, for instance,
like our eye gene, or the
Right.
genes that regulate our eye development are the same, are orthologs of the same gene, which is PAX6.
right. So when you manipulate, no, this is something you do with like unfortunate chicken embryos or something. We're not talking about
mice,
people
yeah.
or mice, right? But when you manipulate PAX6 in mice, you can get them to grow eyes in embryo where they shouldn't be. Similarly, you can get flies to do the same thing when you manipulate PAX6.
And you can switch them.
Right. And so, yeah, that's another thing. You can actually take fly PAX6 gene, put it into a vertebrate embryo, and it will regulate the development of normal vertebrate eyes in that embryo. And so what we think of as these fundamental differences, actually, when you get down to the level of like, how does this organism actually grow itself? They're relatively small changes. And one of the things that means then is that mutations to these regulatory genes that govern embryonic development can result in very significant morphological changes in an adult. And while most of those morphological changes are probably not going to end up being beneficial, it only takes one, really. And at that point, once you have one significantly beneficial morphology change as a result of change to these developmental genes, that can be the thing that sets up an entire new phylum if this happens far enough in the past, which is actually what happened. The reason we have these different quote unquote phyla, because all the named ranks the names of the ranks don't really mean anything. But all these phyla, so-called, that we're getting towards the beginning of the fossil record, when we look at them very early on, they're barely differentiated. So like early Lobopodians, which is another group of stem arthropods, they don't look that different from, the other more
like
arthropodic
stem
things.
onychophorans either.
Right, like stem onychophorans, stem arthropods. I don't think we have stem tardigrades, but whatever.
They're pretty small. So.
Yeah, I wouldn't really expect too much in that area. But yeah, they look much more similar to each other than their, putative descendants do. And the same is true even for chordates. The first chordates we get in the fossil record look like worms. They have weird little tendrils at the front. They don't actually have any bones. They just have cartilage. They're segmented in a way that is much stronger than you're segmented. Because, even humans have some segmentation with things like vertebrae and ribs, but it's much weaker. Whereas these guys have strongly segmented bodies. Mm-hmm. They look like other organisms. And in fact, they even look like embryonic tunicates, sea squirts, which don't even have like a nerve stem. They barely even have a brain. And yet you see these really strong connections, both developmentally, genetically, and in the fossil record. And this is something that evolution predicts, whereas anti-evolution has only ever managed to accommodate these facts. That's the best that anti-evolution has done, is it can say, well, that doesn't disprove my idea. It's like, well, okay, but we predicted. One of the questions I sometimes ask anti-evolutionists is, when was the last time, using your ideas about the history of life, that you were able to successfully predict a new fossil find or a finding from, embryology or evolutionary biology or any of these fields? When was the last time your ideas were used by someone to successfully predict a new phenomenon or a new specimen? And I've yet to have someone actually give me an example that held up to any scrutiny. And I think that's a problem, because science is about, the best you can do in science is successfully make predictions. That is like the absolute gold standard. Making predictions is easy. Successfully making predictions is hard, which is one of the things I've also seen anti-evolutionists get wrong. For instance, Nathaniel Jeanson likes to tout the fact that he's made a whole bunch of predictions. It's like, well, okay, but they've all either turned out to be wrong or you haven't bothered to test them. So get back to me when they turn out to be right. And I think that's one of the very big strengths of evolutionary biology, is it consistently makes predictions that we would not expect to be true except for evolution.
Should I pull up a slide?
Fine by me.
I have a slide with examples. Peter, slide 82, evolution process.
Do you have as many slides as Kent Hovind?
I don't know how many slides Kent Hovind has.
I think he said recently he had a couple thousand.
No.
We'll get on that. Do not let Kent beat you at slide
My computer would not be able to open it. It's too old. These are all...
Well, I think when he says slides, he means physical slides, like that you put through a projector.
Oh, God, no. Why would I have a projector?
Because you're apparently like an ancient old man, like Kent is. Never mind. I'm
My
going to stop now.
computer is old, but I don't have that old technology. Anyway, these are just piggybacking off your point, various fossil predictions.
I love all of these. Because I'll actually talk about a little bit about them. So Microraptor was actually predicted way back in the 19th century, where a paleontologist noticed that, well, actually this time they were just all naturalists, right? Mm-hmm. But this guy was working with ornithology and fossils. And one of the things he noticed is that certain breeds of pigeon developed feathers on their legs instead of scales. And he also noticed that the feathers were asymmetrical, like the flight feathers of a wing. Mm-hmm. And so he proposed, well, maybe at some point in the evolutionary history of birds, they used their hind limbs as wings too. And so they had fully developed feathers for flight, like asymmetrical flight feathers on their legs. And then, lo and behold, here comes Microraptor out of China, which has exactly what he predicted. He said, okay, birds today have 2 wings, but simple mutations can cause them to have what looks like 4 wings. So maybe 4 wings was the ancestral character state for ancient birds. And here we have Microraptor. With Sphecomyrma, one of the things people don't seem to realize, which why would they, but ants are basically just a kind of wasp, right? They're a specific group of mostly flightless wasps. In fact, many of them can still
highly
sting
social
like,
wasp.
It's highly social. But many of them can even still sting like wasps, because a stinger is just a derived ovipositor. And virtually all ants are female, just like virtually all social wasps are female. And so there was the statement, well, we should find at some point in the fossil record an organism that looks like a cross between what we think of as a wasp and what we think of as an ant. And lo and behold, it was Sphecomyrma. With Probainognathus, the interesting thing about Probanognathus is that it actually has two different jaw joints. So it has this sort of stock standard reptile / amphibian jaw, where you have an articular bone at the very back of the jaw. It's tiny, and it rests against the angular and surangular bones, which are bones in the back of most tetrapod jaws. And, so there's a muscle that goes down to the angular and surangular that helps close the jaw, and then the muscle that helps open the jaw is between the articular and the quadrate. But it also has this dentary squamosal joint, which you can see in blue and yellow, right? Sorry, it's very hard for me to read this because it's kind of bad resolution. But anyway, but the thing is, we know, evolutionary biologists knew that it seemed like mammals evolved from what at the time were called mammal-like reptiles. We now call them synapsids. It doesn't matter a whole lot. But most synapsids had this standard reptile-type jaw, and mammals have this very different jaw. So they said, well, at some point, we should expect somewhere probably in the Triassic, because this is where we see a shift from one group to the other, that we can get a synapsid that has both jaw joints. And so before this fossil was ever found, that prediction was made. And then right in the area, and in sort of like vertically, they we expected to find a synapsid with a double jaw joint, which is not something that doesn't exist at all today. We found it. Similarly with Tiktaalik, the reason that the expedition went to far northern Canada, like the wasteland of Canada, where there's not even a permanent settlement for 100 miles, right? Like we're talking remote, which kind of describes most of Canada, if I'm being honest. Sorry, Canadians.
Sorry, Canadians in the chat.
Yeah, I love you. Anyway, but they were specifically going there because they knew that there was the right paleo environment and the right time to find something that was going to fill this gap between relatively basostegocephalian fish and relatively, compared to those stegocephalian fish, relatively derived tetrapods, which is the group that includes amphibians, mammals, reptiles, and birds, if you want to take them out of reptiles. And so that's what they found. They found this organism where it still has a very fish-like tail, but its shoulder girdle has become detached from the skull, which is not the case in fish generally. And it also has these joints in its fins that help give it extra support, but it doesn't quite yet have standard digit. It still seems to have like sort of rays supported by bone. And it has a lot of them, not the standard 5 that tetrapods now have. And so it is exactly both where, when, and with the anatomy that was predicted. The whole reason they even bothered to go into the frozen wasteland. Sorry, I live in a place where it never goes below freezing, and I hate cold temperatures, so I'm very critical of cold places. That's just a big thing.
Yes, me too. And right now I live in a cold place.
My condolences.
Peter, you can take it down now.
Yeah, so that's what I mean. We have this rich history of making a prediction based on the idea that evolution is true. And then lo and behold, we're right. And generally speaking, if you continue to make correct predictions, it usually indicates that the idea you're basing these predictions on is at least somewhat true.
Now, do you think evolution has made predictions that haven't come true?
Absolutely.
Yeah. And generally when that happens, the specific ideas about evolution that predicts have then been discarded. So,
I can give you one off the top of my head.
There you go.
Darwin, so go all the way back to Darwin, thought that dogs were descended from both gray wolves and golden jackals. He predicted, because they didn't have genetic evidence back then, obviously. So he predicted based on the morphological characters that some dogs were descended from golden jackals and some were descended from gray wolves. And now we know, based on genetic and better morphological data, as well as archaeological data now, and even paleogenomic data, that all dogs are descended from gray wolves.
Mm-hmm.
So nobody, is still out there being like, dogs are, some dogs are golden jackals, right?
I'm going to bring it back. Canis aureus for the win.
How about no?
Okay, I won't. But there's another one that's interesting, which is one idea that used to be prevalent in evolution that's now been completely discarded, was orthogenesis. And basically this said that certain lineages could get stuck in a rut. So maybe this group of deer gets stuck in this rut where they just keep getting bigger and bigger antlers. And eventually the antlers just get so darn big that it doesn't work. They just cannot keep reproducing because their heads are just stuck to the ground with these heavy antlers. And this was done to explain certain odd trends that you find in certain fossil mammal taxa, like brontotheres, right?
yeah.
Where they were just
And
getting
deers
what
and
looked like absurd
elephants.
proportions. Elephants too. But it then turned out that no, actually, all of these things were being selected for naturally in the environment, and we don't have any evidence of modern organisms that seem to be stuck in this rut where they continue to evolve in a less and less fit direction.
Well, there was
because
also
of
a
this
degree
momentum.
there was also, to add on to that, a degree of mysticism around
Oh, yeah.
some aspects of orthogenesis, and
Yeah.
since Mendelism came along, those all kind of fell by the wayside. So
Right, but so the interesting thing though is when orthogenesis started to fail to actually make good predictions, when his predictions just didn't pan out, well, the scientific community chucked orthogenesis.
same for ontogeny recapitulates phylogeny. That was another
Right.
really popular one, which also Mendelism did in, because once they realized genes are discrete and you're not turning on like successive sets of like genomes that we have built on top of each other, then
Right.
that just completely fell apart.
Yeah, so yeah, the history of evolution in science, like if you do this, history of science about evolution,
If
you'll
someone
find
to
a
like
lot of really bad ideas. Well, at the time they weren't bad ideas, but ideas that turned out to be wrong, that made bad predictions, and that now are no longer part of evolutionary theory because they were wrong.
if someone were to do an evolution of evolution, perhaps with Dan Stern Cardinal,
What now? That would be interesting, wouldn't it?
that would certainly be interesting. Yeah.
Well,
Wink,
do you
wink.
think there's any type of evidence that could be brought forward that would just cause you to discard the whole theory?
Cambrian rabbits.
I mean, yeah, that would do it.
Because if you find something that's, because the often the problem is, or the objection I hear is of like these temporal paradoxes, is it's like a couple million years earlier than what they originally thought it was, or something like that, and it's like, okay,
Yeah, a few
fine,
million years
great.
in the standard geological
Um,
timescale is
But
fairly
yeah,
minor.
if it's like mammals showing up 540 million years ago at the start of the Cambrian explosion, yeah, that would cause some pretty major quarrels among paleontologists and evolutionary biologists generally.
Also, if we found actual chimeras in nature, so if,
Yes.
for instance, we found like an actual Pegasus, would be pretty
Or
hard to explain.
crocoducks.
Yeah. So, like with a Pegasus, it's like, okay, first, we have no really good explanation as to how a horse would grow an extra limb girdle. Like, we don't have any good way to have that happen. And not only that, but now it's going to grow a dinosaur arm on its mammal frame when it doesn't have nothing about that is compatible with evolution. So if we found an organism like that, and the thing is, every time we find a new organism, it's a potential chimera, right? We could tomorrow find a spider with beetle wings. It's not like ontologically impossible, right? It's not a logical contradiction, but we don't.
I mean, don't we have things like that, such as like we have mammals that lay eggs, right?
So
Doesn't a
actually,
platypus lay
no,
eggs?
that's not a chimera. That is the ancestral character for mammals.
Mhm.
Mammals ancestrally laid eggs. That's
Yep.
why this is an ancient group. That's why genetically they are sister to marsupials and placental mammals. And if
Yep.
you look in the fossil record, then all the pre-Therians were all laying eggs. In fact,
All the Morganeucodontids, the Triconodons.
we even
all
have
those
eggs of
weirdos.
like cynodonts. Yeah, we
Mhm.
know they were laying eggs, and mammals were laying eggs ancestrally. Yes.
I have no idea why a Pegasus would be a problem for evolution.
It's it's
What do
feathers.
people have
It's-
against Pegasus?
Okay.
Oh,
well,
I have
look,
nothing against a Pegasus. I would love to have a Pegasus as a pet.
I mean, but
I'm
like,
just joking because
yeah.
I've heard for some reason I've heard Pegasus brought up in other contexts.
well, it's just it's a popular organism. Everyone's familiar with it. That's just
Yeah.
probably why.
Actually, I think I use it because I don't know too many people who dislike it. I think everyone is sort of like broadly fond of Pegasus. It's relatively inoffensive.
They
No one's
all
like,
watched.
oh, I hate the Pegasus. Get rid of it.
Everybody watched Hercules 1997, and they all fell in love
Exactly.
with with the Pegasus.
Right. It's Like it's one of the nicer of the mythological creatures, because like no one wants to go into like Charybdis, the whirling whirlpool of doom and tentacles or whatever. No one wants that.
Yeah.
But yeah, so the reason it's a problem though is because it's taking. I'm trying to formulate my thoughts. Sorry. So different lineages in evolution, when they split, they stop sharing genetic information between them, right? So one of the things we can look at is the difference between, say, a bat wing and a bird wing. They both do the job of flying, and they have certain similarities, right? Like they both are relatively large limbs with powerful flight muscles attached. But that's basically where the similarity ends. Bat wings incorporate 5 fingers. The first one is relatively short. Then actually the first one is just a thumb, and it's usually free. Then the second finger is relatively short. Then you get a really long third finger, fourth finger, fifth finger, and they kind of turn posteriorly as you go out from digit 1. Whereas birds use only 3 fingers in their wing. The second one, actually the first one is a tiny little nub. Then they get a really long first and second finger, but no subsequent ones. They use feathers instead of skin. And so if you found an animal that otherwise was a bat, but it had a wing from a bird, evolution doesn't have a way to do that, because evolution can't take bird anatomy and copy paste it into a bat. It has to work with the bat anatomy and the bat genome as it is now going forward.
Right. These are phylogenetic constraints.
Right.
Also, Rebecca, I didn't ever ask you for like a time, however long you wanted to go, and I don't want to hold you. I don't want to be rude and hold you too long, because we're almost at the 2-hour mark, and so I don't know if you want to end it now or you want to go.
Yeah, I mean, I think it'd probably be good to wrap it up.
Okay.
But I've enjoyed hearing you guys share your passion. So thank you for.
Oh, my actual passion is flower arrangement. That's not true. Sorry.
I only have one little set of plants, and they're not doing too great.
But Jackson,
They're
your
okay
real
when they're
passion is aquarium. You got a bunch of aquaria.
I have a bunch of aquaria. Yes, they're real passionate about them. Sometimes, like Darwin up there, the little axolotl. I have some African cichlids down there. Have some Xenopus, and then I have some Tetras downstairs. So yes, lots of aquaria. But at any rate, well, Rebecca, I do want to thank you for coming on. Like I said, I didn't say in any emails how long we would be going. So thank you for staying for the 2 hours that you have now.
Yeah, my pleasure, guys.
Yeah, and I want to say thank you too.
Yeah, nice to meet you. Dapper Dino, do you ever show your face on camera?
I have teased that maybe if everyone gets me to a certain subscriber count, that might happen. Generally, I don't. It's not because I'm hiding. It's actually relatively easy to find out who I am if you really want to go digging.
Okay.
But basically, for me, what I do on my channel and in my appearances, kind of attached to the channel, it's not important who I am. All that's important is the science and the arguments. And so my relative lack of showing my face is primarily not because I want to be anonymous, but because I don't want it to become a thing that's personal. Because it's not about me. Like, everyone knows my first name is Timothy. Like, it's not about Tim. It's about some science. And whatever Tim thinks is irrelevant. All that matters is there science to support it, and do the arguments that the Dapper Dino gives work out? So whatever problems you might have with Tim, don't have them with the Dapper Dino.
I got problems with both, sir.
Yeah, that's fair.
And also thank you to Peter for hosting this as always. Thank you, audience, for watching. And I guess we're good, Peter. So thank you, everybody, for coming. Thank you guys for participating. So have a nice night, everyone.
Night. I just want to take a moment to thank my channel members and my patrons on Patreon. They really help keep the lights on here. And I want to thank those pledging $20 or above, especially Bob Knob, Bent Hovland, Ian Chen, Chris Love, Sphincter of Doom, The Venerable Weed, and Patrick Dennis. Without the support of those whose names you see on screen right now, the channel simply wouldn't exist as it does now, and I certainly wouldn't be able to do 2 to 3 videos a week for over a year and a half now. If you would like to help support and make it so that this channel is something that can keep going, There is a link to join below the video, as well as a link to the Patreon in the channel description. If pledging money isn't right for you, please just hit like on this video, subscribe to the channel, and share my videos. That really helps my channel grow.